criterion is to stick around long enough and things start to come your way, you know. So hopefully my voice will carry. You'll be able to hear me on the back. And what I want to talk to you about today is the issue of airway remodeling, in particular with the focus on smooth muscle. So what are the mechanical factors or mechanical determinants that lead to airway narrowing? I'll give myself an arrow here. So you can see the airway here in an asthmatic lung. Oftentimes when these airways are examined, they have this infolding of the epithelium, which is a reflection of the fact that the muscle here in the airway wall has contracted and caused the airway to narrow. Outside, of course, here you have the attachments of the alveoli, the alveolar attachments to the outer wall of the airway. And the factors that, of course, cause airway narrowing, in addition to airway smooth muscle, are the intrinsic recoil or elastic properties of the airway wall itself. And when the airways are small, or if they're narrowed so that the effective lumen becomes very small, surface tension can be significant at the air-liquid interface. And this actually also sucks in the airway, so it acts as if it's an additional constricting force on the airways. And the elements that promote dilation of the airways, keep them open, are of course the long elastic recoil, which is transmitted through the parenchyma to the outer wall of the airway. And the pressure in the pleural space, which of course is usually negative when we breathe, appears to be transmitted in such a way that the pressure outside the airway wall is also equivalent to this pleural pressure. And of course in large airways we have cartilage, which tends to support the airway and prevent its narrowing. Now one of the very important factors, which I'm not going to deal with, this is a sort of a static concept of how the airways work. Of course, when we breathe, we oscillate the airways, we cause oscillatory stretches, and these are very potent in terms of dilating or relaxing the smooth muscle. So that breathing in and of itself is an important dilating factor. However, you can imagine that the quantity of airway smooth muscle here may be important because there are these mechanical impedances to airway narrowing. Therefore, the more muscle we have, the more force is generated, and the more the airways can constrict. And we've known for quite a while, in fact, going back 100 years almost, there are reports of this tissue here, this pink tissue, which is airway smooth muscle, in a biopsy of the airway. So this actually is the epithelial surface of the biopsy. This is some connective tissue under the epithelium, and all of this pink stuff here is airway smooth muscle. So in the asthmatic airway, this is increased in mass, and it increases as a function of the severity of the disease. So in very severe asthma, the muscle becomes very prominent and can actually be virtually adjacent to the epithelium itself. So I'm interested in why it is that this muscle grows, and we call this airway remodeling. There are other elements of airway remodeling, but this is the one that I'm most interested in. Now, you can imagine if you take a biopsy of this sort from a proximal airway by bronchoscopy, you might think that there could be large sampling errors, therefore it might be representative of what really is happening. But curiously, when you take a biopsy like this and you quantify the area of muscle in the biopsy, and you look at it in asthmatics of different severities. So these are severe asthmatics, and we have the mass of muscle here. It's actually the area in a cross-section. And we have moderate asthmatics. You can see that there's actually less muscle in the moderate than in the severe. And if we look at a measure of lung function, the forced expiratory volume in 1 second, this is inversely related to the mass of muscle. So if subjects have a very good FEV1 in the normal range, they're not likely to have much increase in airway smooth muscle. However, if they have a low FEV1, they are more likely to have lots of muscle in their biopsies. So despite the fact that this is just a small sample coming from proximal airway, it does appear in some way or other to be representative of the degree of remodeling that affects the function of the organ overall. Now, you might think, and I certainly did for many years, I thought of our bodies, the human organism, as a sort of a fairly static entity. But of course, we are turning over constantly, and in some ways it sort of surprises me that we wake up in the morning looking like we did when we went to bed. The cellular turnover can be quite large, and this here is an immunohistochemical stain, a double stain, the pink material being smooth muscle in the airway biopsy, and the very dark staining nuclei are nuclei that are expressing proliferating cell nuclear antigen, which is a marker of a cell that is actively undergoing, is synthesizing the DNA and getting ready to go through the cell cycle. So even though muscle, we think of it as being fairly differentiated, has this capacity to turn over as well in the airways. Therefore, when we look at healthy subjects, moderate and severe asthmatics, we find that the number of proliferating cells as a fraction of the total cell nuclei that are present is actually significantly elevated in the severe asthmatics, indicating that even though there is more muscle and it seems to stick around, it's maintained in a new dynamic equilibrium in the severe asthmatic. These are other biopsies. Again, this is from a healthy control. This is a moderate asthmatic, and this is a severe asthmatic. And these numbers here, 0.06, 0.32, and 0.45, are the proportion of the biopsy occupied by muscle. So as we go from left to right, we have increasing amounts of muscle. We also find that the muscle is sometimes well organized. As you can see here, you have muscle fibers that are together in a bundle, but in other areas you have cells that are expressing contractile protein that are distributed in a more random fashion throughout the biopsy, and that also show, in some cases, very modest immunohistochemical staining for the smooth muscle protein alpha-actin. So there appear to be these well-organized, typical smooth muscle cells, but you also have these poorly organized contractile cells that appear in the biopsies as well. Here you can see that you have this punctate staining, which is a reflection of a lot of these cells that are alpha-actin positive, but that are not organized into any bundle. And the term employed by David Ramos when he did the study was non-organized airway contractile elements, because that's exactly what they are. So these increase progressively in the asthmatic as you go from moderate to severe, as well as the total mass of muscle going up, as I mentioned earlier, from moderate to severe. David happened to put a curve through the data points, but again it shows you that there is a relationship between lung function and the mass of muscle in these subjects. So in several studies we found that single biopsy gives us a measure of something that relates to lung function. David also did he did triple immunostaining. He immunostained for T cells, so this brown stained cell here is a T cell that is sitting in the midst of these alpha-actin cells that are smooth muscle. And he also looked for proliferating cells. So he had this triple thing. First of all, we look at the T cells that are present in the airways. He found that they increased from control subjects to moderate to severe asthmatics. So the density of T cells in the tissues went up. So there is a lymphocytic inflammatory infiltrate. And he found also that the number of T cells that were actually sitting within the muscle also went up progressively, although they are, of course, fewer in number than those in the total biopsy, because there are areas in the biopsy here where you have clusters of T cells that are outside the muscle, under the epithelium, but there are T cells within the muscle intimately in contact with it. So the other thing he did was that he looked at the adjacencies of these T cells to the smooth muscle cells, and he showed that they increased as well. Now, previously, and I'll show you his data in a minute, David Ramos had shown that T cells in contact with muscle could trigger the muscle to proliferate in the setting of a culture. So he was interested in looking to see whether there was a relationship between T cells that were present in the biopsies and the number of positive cells that are present. And he found that that there was a substantial increase in these proliferating cells that were sort of juxtaposed to T cells. So now to go back a little in time to sort of explain to you how we have worked on the issue of muscle growing within the airways. We back in the early 90s identified a rat that was the best model of being atopic, in other words, the tendency to sensitize to allergens. This rat distinguishes itself from a whole bunch of other strains in having very high levels of IgE, which is the typical immunoglobulin associated with allergic sensitization. And this animal, when you sensitize it to an experimental allergen such as chicken egg albumin, ovalbumin, and then you expose it subsequently to the ovalbumin, the muscle will grow in the airways. And it doesn't take actually a lot of exposures. So here you can see these are faded pictures from a faded manuscript. And what you can see is this is the muscle area in a healthy control animal or sham animal. And this is the muscle in the airways of the allergen challenged animal. And in small, medium, and large airways, the muscle increases, actually doubles, in response to just 3 exposures to an allergen challenge. So it's very, very plastic. It can undergo this increase in mass very, very quickly. And it turned out that if you actually look at airway function, and the way we usually do that is to look at how easily the airways contract when you deliver a bronchoconstrictive agonist, such as methacholine or serotonin, if you want to use the equivalent of histamine in the rat. So the sensitivity to that drug, to the bronchoconstrictor, was also related to the amount of muscle. The airways, this is a measure of response to the agonist, and that it correlates with, I mean, it's a loose correlation, but nonetheless is there in relationship to the mass of muscle and how these animals react to the agonist. So this supports the idea that the mechanics of airway narrowing that I sort of went through a little bit at the beginning, where the mass of muscle is up against the impedances that prevent airway narrowing, could be an important issue in some cases of hyperresponsiveness or asthma. Now, so how might the muscle grow? So we've been interested in growth factors that might promote that, and for a variety of reasons we came upon the epidermal growth factor receptor, which is of course a major receptor involved in tissue growth, tissue development, and so on. And it has there are kind of a half a dozen ligands for this receptor, and 2 of them here, amphiregulin and heparin-binding epidermal growth factor, are both increased in the airway lavage fluid of animals that are given these allergen challenges. So if you give the animal 3 challenges, and then you lavage the lungs and measure these ligands, you find that they are increased in the animals that are exposed to the allergen. The drug mentioned here, AG-1478, is a drug that will inhibit the epidermal growth factor receptor. So we were also interested in knowing what would happen to the growth of airway smooth muscle if we were to inhibit the effect, this particular pathway that these ligands would cause growth through. And so these are some pictures here again showing you airways. You can see the pink, the red staining is actually muscle in the airways. And this is a high power picture showing you also the quantification of proliferating cells in addition to the just identifying the muscle. So any dark, really dark staining nucleus, best seen here in the epithelium, would represent a proliferating cell. So those cells increase in both the muscle, and those proliferating cells increase in the muscle, and they also increase in the airway epithelium. And the epithelium actually turns into, or has more cells that will produce mucus in response to this particular remodeling pathway. So in small, medium and large airways and in small airways, the muscle increased in mass after these allergen challenges. And the increase was blocked by preventing the epidermal growth factor receptor signaling using this AG1478 compound. So that brings the amount of muscle back down to the control level. And when we looked a week later, the muscle actually in the small airways had increased a little bit more. The muscle in the large airways was pretty much unchanged, although the only significant difference there was between the allergen challenge group and the animals that were treated with the AG1478. So in this particular animal, the muscle does hang around for a while, although we do know that eventually the airways will remodel again back towards normal with loss of that muscle mass. And we don't know how that comes about, and one would have to wonder why that wouldn't happen in human asthma when it is triggered to grow. Yes?
So I saw the other one that also the several red staining of smooth muscle cell. I presume they got probably pulmonary artery smooth muscle cell as well.
Yes, yes.
So did you see any kind of improvement or elimination of the BME of the smooth muscle cells around the vessel?
Yeah, we didn't actually. Yeah, we didn't actually quantify the smooth muscle around the vessel. Mm-hmm. Obvious thing to do. We didn't do that actually, so I can't answer your question. Yeah. Of course, the major surrogate for asthma in all small animals is to measure how the airways respond to methacholine or to an agonist that will cause airway smooth muscle to contract. And we do this by aerosolizing, increasing concentrations of methacholine down the airways, and then measuring the mechanical characteristics of the lungs. In this particular case, the analysis is done using what's called a constant phase model, which partitions the mechanical responses of the lung into large conducting airways, more peripheral airways and lung tissue, and then it also quantifies the stiffness of the lung tissue. So stiffness H is on the right here. The more peripheral lung response, that is a resistive pressure loss, or a resistance. In other words, pressure losses related to flow. And here we have pressure losses related to flow, but in large airways. So what happens is that after the animals are exposed to the methacholine, they do become hyper-responsive. The airway reaction is larger, as measured by all of these parameters. And some of these parameters are brought back down towards control levels by treating the animals with this 1478 compound. Now, we also did what's called laser microdissection and capture. So this is a picture of the airway here, and the muscle layer was basically cut out using a laser, and this shows you what you can recover. And then this muscle tissue can be analyzed for gene expression. So when this was done, we found that myosin heavy chain, which is one of the important contractile proteins, was significantly increased in expression following remodeling. and this was prevented by the inhibition of the EGF receptor. That's a little bit counterintuitive because people have believed that muscle either is a contractile tissue or when it proliferates it loses its contractile proteins. But it doesn't seem that in vivo in this particular model this happens. That idea of a sort of a range of phenotypes, which Andy Haleko actually has done a lot of work on, is a kind of been shown mainly in culture, but in the intact animal here it doesn't appear that that particular idea holds up. So we have a model where we can challenge the animal, we can get muscle to grow. It seems to have physiological implications in terms of increasing airway hyperresponsiveness. And I'm now going to go back to the T cells that I mentioned at the beginning. So we've been interested, as I say, in the T cell for quite a long time because we believe that this is the major driver of the of asthma in its many forms. So in this experiment, what was done was that the brown Norway rat was given sensitized T cells from a donor animal. So the animals that were challenged were actually naive, but they were given T cells from a sensitized donor animal. Now the pictures here show you, this is a muscle cell. It's shown in green. And the T cell is shown in red, and this is a T cell sitting right on the muscle cell. So in this animal as well, you can find T cells in intimate contact with muscle. Now, in this particular case, this T cell was one that was labeled before administration. So it was actually transduced to express EGFP, a fluorescent protein. And that protein, of course, then can be identified in the intact animal's lung after the harvesting of the lungs from the animal. And this here is one of those cells. So we know that the T cells that were administered to the animal, actually some of them made it into the airways and some of them made it actually right onto the muscle. The muscle grew in these animals that were given the T cells and challenged 3 times with ovalbumin. So the T cell and nothing else was able to transfer that characteristic response to the recipient animal. And here you can see in the muscle illustrations of proliferating muscle cells, and when they were all counted, in the smooth muscle they went up very considerably following the allergen challenges. Proliferating cells also went up in the epithelium. As I mentioned, the epithelium undergoes remodeling as well. Now, what was then done was to take the T cells, isolate the T cells, and to put them into culture with CD4 positive cells. And these diagrams here are showing flow cytometry using a marker of cell proliferation, which is this bromodeoxyuridine incorporation. So this bromodeoxyuridine is incorporated into the DNA, and so when the DNA is synthesized, you can stain for this particular marker. So what we have here then are CD4 positive T cells that are oversensitized and are put in direct contact with T cells. And we find that there's a cloud of cells expressing or showing staining for bromodeoxyuridine. In other words, they have incorporated the RDU, and so they are proliferating. However, if we put the cells in a transwell, so they were the medium in which the T cells on the muscle were sitting, this medium could, of course, allow diffusion of soluble factors, but it didn't allow contact of the cells. And under those circumstances, the proliferation rate of the cells was the same as that of the control cells. So the presence of the T cells with the muscle can cause the muscle to proliferate on condition that the cells are actually in contact, direct contact. And if the T cells are in direct contact, but they haven't been previously activated through exposure to antigen, they also do not increase the proliferation rate of the muscle. And this is just a sort of dose response of T cells on the muscle. The more you put on, the larger the proliferation is. Sorry. Unwanted interference from Skype. So we've gone on further with this, and we wondered whether the mechanism by which the muscle grows in vivo in the animals, which is mediated by the epidermal growth factor receptor, whether this could actually be implicated in this T-cell driven smooth muscle proliferation. So these experiments here show smooth muscle on its own and smooth muscle in co-culture with T cells. And the green staining that you see here is fluorescence staining for the active EGF receptor. So the phosphorylated EGF receptor shows in green here. So the co-culture causes the muscle to grow. Bromodeoxyuridine incorporation increases. If we use this AG compound, we can bring down, we can bring down the proliferation. And the AG compound on its own does very little to the baseline proliferation of the cells that are just exposed to some fetal Brovin serum. So it looks like a similar mechanism accounts for the proliferation of smooth muscle in vitro in response to T-cell contact as it does in the intact animal. Now, we actually have nailed it down to one of the ligands, heparin-binding epidermal growth factor, which I mentioned to you earlier, appears in the lavage fluid following an allergen challenge. So this particular ligand for the EGF receptor is markedly upregulated when you put, so this is the RNA for it, when you put the T cell in co-culture with muscle, and now you isolate the T cells and you look for the expression of this ligand, you find that it's markedly upregulated, like 150-fold upregulation following a contact between the T cell and the muscle. So we know that T cells do actually express growth factors, but we know very little about what their function is. My idea when finding this was that T cells are not only immune effector cells, inflammatory cells, but that they could also possibly reconstitute the tissue that's damaged by an inflammatory process. It would seem to me logical that if you're going to have inflammation that may cause tissue damage, that it would be nice to couple the magnitude of the inflammation to the repair process, and this is a potential mechanism whereby that could happen. And if we neutralize the effects of heparin-binding EGF by adding a neutralizing antibody to the co-culture, thank you very much, you're very kind. The proliferation, the enhanced proliferation of the muscle goes away. So it does appear that this actually is the responsible ligand for the EGF receptor, causing the muscle to grow in the context of a T-cell co-culture. So I've mentioned to you then several times the importance of this contact between the muscle cell and the T-cell for this phenomenon to happen. And from here on it kind of gets weirder and weirder. But you can't see this terribly well, unfortunately, but it'll kind of set us up for the next couple of slides. So what we have here is a T cell, indicated by the arrow, and next to it is a smooth muscle cell. If we add an antibody to this adhesion molecule here, CD44, and if we apply attraction to the T cell, we can pull the T cell away from the muscle, provided we neutralize this particular molecule here. So hyaluronic acid and CD44 will bind together, so it's kind of a glue that's sticking the T cell onto the muscle. And it seems to be the principal adhesion mechanism, although there are other adhesion molecules involved. And you can see that the arrow is getting farther and farther away from the muscle, so the T cell is actually being pulled at quite a distance from the muscle once we have this antibody on board. And when you count the number of cells that can be detached, if you add the antibody before the co-culture, you can detach them all quite easily. if you add the antibody and wait a half an hour, about half of them can be detached by pulling on the cells. Now, how did we pull on the cells? We pulled on them using a laser tweezers. So the laser tweezers uses laser light that is shone into the medium, and that creates an energy well, and you can move the laser, and in this way you can actually exert attraction on the cell. You can kind of capture the cell, I guess, in the energy well and try and move it. So this is another picture here of a T cell sitting on a smooth muscle cell. And so we're pulling on it now with the laser tweezers. And we were very struck by the fact that that cell popped right back to where it started. That was a very surprising observation to us. Here you can see a similar thing. It's not as nice a picture, but again, you're pulling on the T cell. When you pull it up and let go, it goes back down as if there is a string attached. So when we look more carefully at these images, we saw that there seemed to be something attaching the two cells. So this, the quality of the images in this laser trap device is not that great, but they looked like there was something there. So, you know, we did what we should have done before, which is to start to read the literature, and we found that people had actually described tubes connecting immune effector cells before. So they described in T cells that you could have these narrow tubes that would interconnect with the T cells. These tubes were described in these original papers as nanotubes, but I think in many cases they're closer to microtubes than nanotubes. But I think that there was a time when you called something nano, you know, you had a better chance of getting your paper in a big journal. So we had a look at these structures in a variety of ways. These are some images obtained using atomic force microscopy. So here you're touching the cell and creating an image of it with the atomic force microscope. This is a smooth muscle cell in culture. And these are T cells that have been placed in culture with the muscle. And you can see that some of these T cells are putting out these large, fairly large and long protrusions that are coming into contact with the muscle. And here we can see one at different scales. So it seems to be inserting into the muscle membrane. Now these are some other images, confocal images. Here we have a T cell, and you can see that there's little tiny projections. It's the same image over here, these tiny, narrow, thin projections. And then there's this larger projection which kind of seems to fan out and have a sort of foot process at the end which gloms onto the muscle. And in fact, in some cases, if the T cell retracts this process, sometimes it pulls away a piece of the muscle membrane. And apparently this is also a described phenomenon called trogocytosis, where cells actually decide that they're going to cannibalize a piece of another cell. So these are these tubular connections. These are other images. Again, the cells are stained with calcine so that we can see them nicely. So this is a T cell with a very long projection, and then this kind of foot process at the end here. And this is a T cell, a muscle cell over which it is it's traversing. So this one also is putting down a process that is joining the membrane of the muscle here. Some scanning electron microscopy images showing you a T cell that is this very long process, and again this curious sort of foot that it has here, and the underlying smooth muscle. This is a highly magnified view of this little area here, and this is a magnified view of this area. You can hardly see those tubes. So these are truly sort of nanotubular structures that are put out and attached to the cell membrane of the muscle cell. Again, some of these other images of the same phenomena, a T cell with this tube, and it kind of goes under the cell with this process that widens out. So these tubes, so this here is a T cell, and this is a tube which is large initially and then narrows down. They contain actin, so the cytoskeleton that is present in the muscle here. Here you see stress fibers that are actin, but here you also have actin in these tubes. And they also have tubulin. So this is a T cell with a muscle cell, and it is stained here for tubulin, and you can see that it's positive for that. Over here it's stained for calcium, which is a green fluorescent marker to identify the fact that this is a T cell. So tubulin is important for, is obviously a part of microtubules, and these microtubules can be involved in the transfer of organelles from one cell to another. So there is a possibility, in fact there's evidence, and I'll show you some, that the muscle or the T cell might transfer organelles from one cell to the other through these tubes, which opens up a whole range of questions about how two cells might relate one to the other, and what materials they might transfer, and how they might influence the phenotype of that other cell. I'll get back to that in a minute, but we were interested then in what were the potential mediators of the tube formation. So what was it that the smooth muscle might produce that would encourage the T cell to generate these tubes? And we thought, well, they look like the sort of the dendrites of neurons, so we thought if we look at what's known about the formation of a sprouting of neurons, we might get some clues. So we screened a number of these molecules, and the fibroblast growth factor family, which is quite large, one of them popped up as being overexpressed in the muscle when it was co-cultured with the T cell. So we focused our attention on that. And we looked to see if both the ligand and its receptor were expressed on the cells. This is a T cell. This is a muscle cell nucleus here. And we looked, as I say, for the expression of both the ligand and the receptor. Here we're looking at the gene expression of the receptor in the muscle. So the receptor didn't actually change, for this ligand didn't change in the muscle, which is not what we were all that interested in, but we did it as a control. But in the T cell, the receptors for FGF2 were very markedly upregulated. So if a ligand is produced by the muscle and the receptor is upregulated in the T cell, then this kind of is sort of in line with what we had been hoping to see. So we were able to find that FGF2 was synthesized by smooth muscle. If we took the cells, lysed them, we could measure it. Now, with a lot of these substances, depending on their chemical characteristics, they may be more or less easy to measure. And FGF is a very basic protein with an isoelectric point that is, I think, somewhere around the 10 or 11. So proteins like that stick like mad to plastic, to glass, and to pretty much anything that you're using. So they can be hard to measure in the supernatant of cell cultures, but there's enough in the cell lysate to be able to measure it. So we were able to show that muscle had that ligand. So the next thing we did was that we counted the nanotubes. So we had the co-cultures, we counted the tubes running from the T cell to the muscle, and we looked to see what inhibitors of this pathway would do. So these two PD compounds are actually pharmacological inhibitors of the FGF receptor, and in each case the number of nanotubes came way down following the treatment. This is an inhibitor of the EGF receptor, and that also, and presumably by different mechanisms or independent mechanisms, brings down the number of nanotubes as well. So we think we now have a handle on what it is that the muscle is producing to encourage the T cell to make these tubes. We inhibited FGF2 synthesis in the muscle by using an siRNA. And when we did that, it brought down the number of nanotubes that were formed between the T cell and the muscle very markedly. If we added back FGF to the mix, we could actually rescue the phenomenon. When we added an antibody to FGF2, so presumably it is actually secreted, goes from the muscle extracellularly to the T cell and encourages that formation of nanotubes. When we neutralize this, we also were able to bring down the number of nanotubes that were formed. Now, so I mentioned earlier the business of these nanotubes being able to exchange material. In this case, what we have is pictures of cells that are stained with two lipophilic dyes. So these are basically staining the cell membranes. And we stain the T cells in green and we stain the muscle in red. But following a co-culture, you find that both muscle and T cells are staining green, and both muscles and T cells are actually staining red in addition. So there is this exchange of membrane between the T cells and the muscle. And here this shows sort of a co-localization of the two dyes, showing you that you have plenty of areas where both dyes show up together in significant amount. So membrane is probably flowing from one cell to another as they make contact, which means that they might also exchange a lot of surface molecules that could alter the function of one cell. You can imagine that you might have the CD4 marker on the T cell that might end up on the muscle. Kind of interesting to know when you get hybrid characteristics, you know, how do these cells change their behavior subsequently. Now, I mentioned earlier that the CD44 was an important way in which the T cell and the muscle stuck together. We found when we added antibody to CD44, as I mentioned, it allowed us to detach the T cell from the muscle very well. In addition to that, however, when we stained for apoptotic cells, so cells that were dying by apoptosis, these kind of very black staining cells here are actually apoptotic T cells. And we counted those cells and we found that in the presence of this antibody, the number of apoptotic T cells went up and the number of live T cells in the co-culture went down. So it seemed then that the T cell was living in greater comfort, survived better in the presence of the muscle as long as it was allowed to be in contact. So we wondered, you know, could factors that assist the survival of the cell be transferred up those tubes? And we looked at two of these factors that are so-called anti-apoptotic factors. One is BCL2 and the other is MCL1. And these are proteins that normally have an action to enhance cell survival. So we found that in fact both BCL2 could be identified in the tubes. There's this little punctate staining here. And we also found that MCL1 also could be seen within the T cells and within the muscle and within the tubes. Here you can see a tube where it is present. Now, we actually transfected the muscle cell so that it was actually generating a fluorescently labeled MCL1 and BCL2 for these experiments. And so, in fact, what we're seeing in the T cell is MCL1 that has made its way from the muscle into the T cell during co-culture. And I'll just go, so this is another illustration of that phenomenon where MCL1 is present in the tube and is present in the T cell following co-culture. Now, we actually knocked down MCL1 synthesis by the muscle using a silencing RNA, and we knocked down BCL2 as well. And we found that BCL2 alone being knocked down did absolutely nothing to the rate of apoptosis of the T cells, but knocking down MCL1 increased the T cell apoptosis. So it seems that this particular factor is transferred from muscle and is accounting for how the T cells survive better in the presence of this of the smooth muscle cell. Now, this is an illustration here of the mitochondrial network within a smooth muscle. So mitochondria, which are obviously the source of energy for these contracting cells, are abundant in the smooth muscle. You see this very rich network here. And this here is a cute picture of a T cell that has a mitochondrion sitting in its trunk. I mean, this is the tube here, the tubes they make, and there's a mitochondrion sitting in there. Here we have a whole cluster of mitochondria that seem to be sort of clustered around the entrance to that tube, making you wonder, you know, which direction they're actually going in. So what we did was we labeled the mitochondria in the muscle by, again, transfecting the muscle with a protein that marked the mitochondrion. And we found that we could find the marker of the mitochondria. We could find marked mitochondria, in fact, within tubes going to the T cell. So this is a T cell, this is muscle. This is the tube between them. And if we look here, we see the red staining, which is the marker for the mitochondria in the muscle. So mitochondria are actually passing through the tubes from the muscle into the T cell. We've shown that by flow cytometry, where you actually take the cells and look for the stained mitochondria in the T cell following co-culture with the muscle. And we have a cluster of these cells here. And in fact, about 10% of the T cells had mitochondria from the muscle after they had been put together. So there's obviously this machinery that can do that. Now, we wondered whether this affected the oxygen consumption by the T cell. And in these experiments, actually, as luck would have it, it actually went the opposite direction. So with co-culture, this consumption of oxygen here, which is measured by a device called the Seahorse, which is it's a 96-well plate-based assay system where you have an oxygen sensor that is put down into the medium, and a microchamber is sort of created by the sensor going down and sort of trapping a small amount of fluid over the cells. And then the rate of oxygen consumption can be measured in that very small space. And also the appearance of hydrogen ion can be measured, which is what the second graph is showing. And it looks actually, following co-culture, like there's less oxidative, there's less oxidation and less hydrogen ion production by the T cell. So it's actually possible that the T cell is transferring more mitochondria than it is receiving, because we've only looked at the unidirectional flow from muscle to T cell. It may be actually, the net flow might be greater in the opposite direction. So we need to try and do that, but it's not as easy. So I'll conclude actually by saying that airway smooth muscle remodeling is, we believe, an important part of the pathogenesis of asthma. It won't account for airway hyperresponsiveness or reactive airways in all asthmatics, because not all asthmatics have much in the way of remodeling of the muscle. But the severe asthmatics certainly have an increase in mass, and I think that on mechanical grounds it is likely that that muscle will cause more airway narrowing and contribute to the disease. Now, the T cell certainly infiltrates the muscle in vivo in asthmatics. It does so also in these animal models, and it's in very close contact with the cells. And I suspect that it is talking to those cells in vivo, perhaps in ways that we've been finding when we've looked in vitro. It's a lot more difficult to actually address these questions, obviously in vivo. The epidermal growth factor receptor, I think, is a very viable candidate for this remodeling process. But of course, the drugs that are used for this are chemotherapeutic drugs, in fact, and not without their side effects. So it's not a readily applicable treatment for asthma at this point in time. And I think this whole sort of nanotube, microtube story is sort of a fascinating area of biology that's going to be of interest over the coming years as we understand how cells communicate. I mean, this has been known for bacteria for a long time, that they can communicate and exchange material, but much less so mammalian cells. And I think that there is an emerging literature. In fact, the numbers of papers on the topic is sort of rising fairly rapidly. And I will leave it at that, and thank you very much. I hope I didn't bore you all to death with this. Biology is not very bioengineering, but maybe somebody would be interested in the rate of movement of organelles up the nanotubes and try and model it and understand the motor machinery that's involved. Thank you very much. Yes.
How applicable is the mouse model to the human scenario? And the follow-up to that is then, is there a human pathology where there's reduced T cells that would perhaps produce a reduced incidence, population incidence of asthma?
Yes. So both very pertinent questions. So the first one is like how pertinent is a mouse model to human asthma? All of our studies, all of our intact animal studies have been done with rats actually, although we do work with mice. The rat has an airway architecture that is, I think, more akin to larger mammals because it does have a bronchial blood flow, and it has a comparable architecture. I think the immune aspects of asthma are very well reproduced in animal models because they are very archaic. The immune system goes back hundreds of millions of years. It's interesting in relationship to the natural experiment with regard to T cell diseases that take T cells out of the picture. And the first one that we were interested in was, of course, during the AIDS epidemic, when we had subjects with very diminished CD4 positive T cells, and they actually have more allergies and more asthma than you would anticipate. So this is where you have to look at the different kinds of T cell subsets. You have the T cells that deal with allergic inflammation that were related to our defenses against parasitic infestations in the past. And that part of our immunity, even though it can be very nicely knocked down with corticosteroids, which is why they're useful, is however very robust, and it withstands the immunosuppression of the AIDS virus, where it's the other kinds of inflammatory T cells that get knocked out of the picture. And when we give immunosuppressives to prevent rejection of organs, for example, renal transplants or whatever, it is also that allergic side of our immunity, the so-called Th2 side, that is persistent. So I think there's nothing irreconcilable between the observations of these kinds of natural experiments with what we know about the T cell. There's just many subsets of T cells that can contribute in different ways and that reciprocally regulate each other. As for the mouse, I think the mouse has a very thin airway, which is nourished from the alveolar capillaries, doesn't have a bronchial circulation. So I think there are likely going to be differences related to that architecture. Also, there are issues with the size. The size of the airway in the mouse, I think, will obligatorily require surfactant to keep those airways from wetting and forming, liquid bridges and collapsing and so on. And larger mammals probably don't need, they're not so dependent on surface stabilizing, surfactant materials to stabilize the airways. So it's possible that in the mouse, disruption of surface tension might actually be a major issue that would distinguish it from other animals in terms of its reaction. There was a question back there.
So excellent talk, thank you. So I had a question talking about the nanotubes that are formed between the T cells and the airway smooth muscle. A little bit of context, I'm a PhD student with Andrew Colbeco, and I'm interested in finding out, see whether or not these nanotubes that are formed between the two cell types, does that only occur within the contractile or the proliferative state of the airway smooth muscle cells? Or is there a difference between the two in terms of formation of the tubes?
Yeah, I don't really know. I don't know. When we have neutralized when we've knocked down the nanotubes, loss of, or reducing the numbers of nanotubes has not affected the proliferation rate of the muscle in contact with T cell. So we're still, I don't know, that is a contact dependent thing, but may not require the tubes. On the other hand, when the cell is sitting, when the two cells are sitting on each other, you might have an extremely short nanotube that you can't identify microscopically, you know, so the actual detection techniques are not optimal. I'm not sure, based on our earlier, I'm not sure when you came, but the rat experiments we did where we challenged the animals, we had an increase in mass, lots of proliferating smooth muscle. We pulled out the muscle by laser capture, and we found that the myosin heavy chain was increased in expression, making me wonder whether, in fact, proliferative and contractile are actually opposite ends of the spectrum, or whether you couldn't have the two operating together. Now, you might think that transcription factors would turn on one, turn off the other, but I'm not sure. I think it needs to be shown in vivo as yet, you know. But I'm not sure that a particular kind of cell has a predilection for creating the nanotubes. It's an interesting point. I don't know.
Are you going to follow up further experiments with the oxidative stress that is occurring between T cells and the airway smooth muscle for transference of the mitochondria?
Is it related to stress, you said?
Are you going to further look into oxidative stress between
oxidative stress?
Because you're showing the mitochondria going across.
Yeah. Right now, I just wanted to understand what the flux of mitochondria was, the net flux, whether it's actually more going out of the T cells and into the muscles, vice versa. To understand that first, I hadn't thought about the issue of oxidation per se and what that might do. No. I mean, we have looked at oxidative stress in like whole animals, and it is important in the airway dysfunction, but I haven't thought about it yet. Yes?
You first.
Both.
I already
Okay, go
asked
ahead.
one. So, Jim, I have a question about, as you know, there is a mast cells in the muscle bundle in the airway. So, I'm just thinking where the transfer of membrane between mast cells and airway smooth muscle cells can explain a little bit some controversies in the literature in terms of receptor expression, what the muscle express, what other cells express.
Yeah, that's right. I mean, it's a good point. So you would be wondering about like IG receptors, for example, whether they could migrate from the muscle, from the mast cell onto the muscle. I see no reason why not. I mean, it seems to me that once the membranes come into contact, I'm not sure why there wouldn't be flow of the material from one to the other. I mean, I think that our cartoon concepts of cells, you know, misleads us, you know. I mean, for the longest time, we've looked at pathological specimens and they're static, right? You think of everything in a kind of static manner. And I think that things, you know, we have to do a lot more dynamic imaging and so on, live imaging, follow the time course of events to sort of understand a lot better what's happening. But have you found that you could find the protein but not the RNA or something of that sort? Is there?
Yeah, we found both.
You found both, yeah. Well, the RNA might also be transferred. Might well be transferred. Yes.
So I'm thinking about the T cells.
About what, sorry?
The T cells.
Yes.
In not only animal but also in human, if you get more T cells sitting on smooth muscle cells on the airway, it must come from the blood. So asthma, as you said, I'm not an asthma person, but it's inflammation kind of process and remodeling process, and people are looking at eosinophil and neutrophil, so on and so forth. You're looking at T cells. Is there any kind of like non-neutrophil, the neutrophil or eosinophil have the similar kind of communication with smooth muscle cells?
Nobody's looked for that. The cells have been looked at, or sort of dendritic cells have been looked at. Some of the work has been done on like cancer cells, like pheochromocytoma cells. Mesenchymal stem cells have been looked at, and somebody has suggested that they transfer mitochondria to the airway epithelium in a mouse that is exposed to cigarette smoke. Mitotracker is a tracking marker for mitochondria that is used a lot, and we find that it actually in the culture medium it leaks out, even though you don't see it. It leaks out and is taken up by other cells. So